My name is Zach Smith. I'm a senior application scientist with Beckman Coulter. I work on automating NGS sample workflows, or library prep workflows on our automated liquid handlers. And I wanted to give you just go through a few minutes today to talk about how to handle all the data that's coming off of all these workflows that you're generating in the lab and how the problem can be managed with our Dart software. So just briefly, we're going to talk about the problem of data management in the laboratory and discuss Dart 2.0 as a data management solution, and briefly go through how you can integrate Dart into an NGS workflow. So your general NGS workflow will start with accession and plating of various samples. You might be consolidating samples from more than one project onto a plate for library prep. You could then work on if you're starting with whole tissues, then you potentially would need to extract those nucleic acids, either DNA or RNA. We're using our Sprite technologies, so it would be RNA advanced for RNA, DNA advanced for DNA, GenFind, and several other products that we offer for extraction of nucleic acids. And then you would need to quantify and normalize those and move on to library preparation with any number of reagent vendors that offer next generation sequencing library prep kits, including Illumina, NEB, Swift, and Rubicon and Kappa, and then move on to sequencing. So data can accumulate rapidly during these sorts of experiments, so you have to merge your sample data onto a plate and record everything associated with that sample. Merge the sample ID with reagent lots during nucleic acid extraction. Also, you've got to keep track of your quantification and normalization data. I mean, what is the actual concentration of the nucleic acid coming out of extraction, and what you're actually going to normalize the sample to. Then merge all that data again with all of the information from the NGS library preparation kits. So this would include lot numbers of various reagent components from the kits, and then exporting all that data to Illumina, the Illumina experiment manager for actually running the sequencing. Also exporting that data to your LIMS system for downstream analysis so that your bioinformaticians know what samples they're working with and what bioinformatics analysis they can perform on those samples. So as you can see, you can accumulate an awful lot of data even on one particular sample well. How much volume how much volume was in the well, what was the concentration of the sample, am I working with avocados or am I working with human samples? There's all kinds of sample data that can accumulate during the course of this experiment, and it only gets worse the higher throughput you become, particularly when you're talking about an automation solutions like the ones we offer that allow you to do even more throughput through your laboratory. So, DART stands for Data Acquisition and Reporting Tool. It's a software package that we have developed to work with our Biomech liquid handling software. It also incidentally works with our SAMI process management software. And DART is a number of things. DART is a collector of information, so it gathers up all of the information that occurs during the course of a run. So Biomech has two different ways of handling data. One is data sets. So these are, it's a native function of Biomech software that uses arrays to capture any sort of data you can imagine. Most often it's volumes, but you can put sample ID numbers and whatnot into data sets. And there's also the log files that the instrument generates when it's doing the actual liquid handling part of the process. So DART is able to sample all of those sources of input from the Biomech. DART's also a repository, so it works directly with Microsoft SQL database to accumulate and store all that data. It cannot be removed from the data from the DART repository. And it's also a reporting tool, and this is one of its better functions is that it goes out and it allows the user to browse, query, create reports, and keep track of all that data in real time. So DART actually interfaces with a number of different points along this workflow. So you can actually import data directly into DART, and I'll go over a couple ways of doing that here in a minute. But it can also take data from associated devices. So if you have a plate reader attached to your network, you can take your quantification data from your plate reader and dump it straight into DART. It also captures all the information from the Biomech liquid handler, and then it can use that information to feed straight into the sequencer for setting up your sample sheet. So, as I said earlier, Biomech has a function called datasets, and there are 4 dataset steps that come natively in the Biomech software. We can create datasets, we can copy, we can delete them, we can perform mathematical operations on them and report the datasets to a flat file. The biomech steps you're seeing with the captions are in blue are all the additional biomech steps that come with the Dart package. So these allow us to load labware data from the Dart repository, add or modify that data, create Dart reports during runtime, and I'll explain how that works in a minute. You can actually modify the script on the biomech to test to see if a data set is available. If it is, then do certain actions on the liquid handler. If it's not, don't do those actions. You can create new data sets de novo, or you can execute different steps when validating a method versus runtime. So this will help you when you're doing method creation to help debug. So there's a couple different ways of getting data into the Dart repository. One is the data importer. So you can actually accession sample and plate data without actually launching a biomech run using this part of the software. And allows you to just bring in a comma separated or a tab separated value file, like what you generate from your LIM system, to give you the plate barcode name, the labware, the index that might be associated with that, sample IDs, which PI it was, the sample was from, so on and so forth. The other way is actually just using the biomech. So biomech will actually update, or Dart will actually capture information from the biomech during the course of a liquid handling run, and using the biomech log files and any data sets that were actually in that run to upload into the Dart repository. So the other thing about Dart is Dart reports out, and it reports out in a couple of different ways. Dart can report out based off of CSV formatted or text tab delimited format reports that are generated actually by the biomech during the run. That's one option you have. You can also access Dart via any web-capable device. So this could be your smartphone, this could be your laptop. As long as you're capable of launching a browser window, you can access the Dart. You can access Dart. So the reports that Dart generates are built off of the Dart Query Builder. So the Query Builder allows you to include any column that you want in a report. You can create reports on runs, on particular labware, on different wells if you want to. You can create it in whatever format you desire. So if your LIM system requires a certain format in order to get data into your LIM system, Dart allows you to change those formats fairly readily with the Query Builder. And it's really quite interesting. One of the things you can do with this is it actually works with Excel to automatically refresh your reports. So if you do a run and you have a report pre-configured, all you'd have to do is launch Microsoft Excel, hit the refresh all button, it will automatically query the database, the Dart repository, and will grab all your information and port it into the files. So it's actually fairly straightforward. It doesn't require an IT guy to make sure your pipeline's up and running. So this is just a look at the Dart live data browser. So the live data browser is just a website that you can go to with it, like again, any web-capable device. So this could be your smartphone, this could be a desktop, where it allows you to look at particular labware. You can actually see if a biomech run has completed, so that if you go ahead and set the biomech up and have it do its thing, you can go off and get some coffee while it's running and keep an eye on it. Or you could do more productive things like, science. You can look at method histories. If you're a lab manager, you can actually get run rates on consumables. You can find out if you have multiple instruments, which ones are being used in your lab the most. If there's a particular biomech that your technicians are favoring versus one other biomech, why are they favoring that? Is that because the method only exists on biomech 1 versus 2, or potentially is it time to schedule a service visit? There's a number of things you can do with this. It's a great way of keeping your lab manager sane and stable and employed, so it's a good thing to have. So I created this schematic just to illustrate various points where you could work Dart into a fairly lengthy NGS protocol. So this is the Illumina Nextera Rapid Capture method. So on the top row, we're going to have the actual part of the workflow in various bins. So we'll rearray the genomic DNA, we'll do the tagmentation and actual library preparation steps in this bin. We'll quantify, normalize, and pool the libraries, do the hide capture part of the workflow, and then normalize and pool the libraries post-capture for sequencing. So each one of these boxes represents a separate biomech method that you could build, and then the locations where you see in and out of here is where Dart would be moving information in and out of those various biomech methods. One of the things that's very handy about Dart is you can move information seamlessly between biomechs. If you have multiple biomechs in the lab, you can move data between methods on the same biomech seamlessly over time. so you don't have to worry about manually inputting data into your liquid handler if that's what if you've got it configured. So this is just an example of some of the data you can expect to get out of the query builder with this particular method. The one that I find probably the most intriguing is this one here. So I don't know how many of you have worked with Illumina Experiment Manager, but it can be fun to keep adding rows and rows and rows. One of the things Dart can do is it can actually report out all of your I5, I7 indices in a format that's ready to go straight into Illumina Experiment Manager, and hopefully cut down on any transcription errors that might result in moving sample ID numbers and whatnot. So and with that, I'd like to thank John Snyder, Andrew Eckert, and Clint Davis, and I'm Zach Smith. And if you have any questions for us, please come see us at booth 9 or our posters at 11 and 12. So thank you very much.